Hello friends, this video on biomolecules part 25 is brought to you by examfear.com. No more fear from exam. Secondary and tertiary structure. Primary structure, secondary structure and tertiary structure for the nucleic acid. Right? If you see here, what is happening here is these are my nucleotides, right? Nucleobases. This whole thing is a nucleotide. Actually, these are my nucleobases. This is my backbone. That is my sugar and the phosphoric acid, and this is my nucleobases. So we talk about the primary structure. Primary structure is nothing but the sequence of letter that indicates the nucleotides. Example AUG, right? GAC, all this, and three of these will form one codon. Three base will form one codon. So primary structure is nothing but sequence of letter that indicates order of nucleotides. So in DNA, we'll use the GACT in DNA. For RNA, we use sequence of GACU because T is not used, uracil is used. Right? So this sequence, so primary structure is nothing but sequence of nucleotides. Because each sequence will represent one information. And these sequences is responsible for unique character. And this has a direct impact on the functioning of living organism. Right? Because, and this sequence also help in finding the parents of a kid, because a parent will pass on this sequence to the kid. Almost the same. So with that, if there's a match, they can say, okay, this kid belongs to this parent. So primary structure is nothing but the sequence of nucleotides. So once I have got the sequence, then I need to form the secondary structure, right? And this is for DNA, it is a double strand helix structure, and Jane Watson and Francis Crick gave this structure to this DNA, double stranded helix structure. So if you see in DNA, there are two nucleic acid chain are wound about each other and held together by hydrogen bond. So you see, this is one chain. This is one chain, this is another chain, chain 1 and chain 2. Right? These two chains are bound to this hydrogen bond between these. Right? Between this between this pair of bases, we have a hydrogen bonding, and there's only certain pairs which complement to each other. For example, my guanine will form a bond with cytosine. If you see, green and blue will form a bond. Here you see green and blue. Similarly, adenine will form a bond with thymine. If you see, yellow and red will form a bond. Yellow, red, yellow, red. Yellow, red, yellow, red. So green forms bond with blue and yellow with red. That is the pairing, right? So they are these two strands are complementary to each other. Actually, they are complementary because they form a hydrogen bond between specific pairs only. So H bond between specific pair only. Example, G will form with C, A will form with T. Correct. So once I've got the secondary structure, but if you see, to get the exact precise 3D structure, we have to take into consideration the geometrical and the steric constraint. So then comes the tertiary structure. Here if you see, there are so many secondary structure combined to form a tertiary structure. Right? When you take into consideration all this geometrical and steric constraint, you talk about a tertiary structure. We don't have quaternary structure for nucleic acids, we have only tertiary structures for nucleic acid. Thank you. Visit examfear.com to watch more videos, attempt free online tests, get free study materials, find tutors and mentors, and much more. Thanks once again.